Hi learners, how are you all? Today we will be learning amino acids in this video. Yes, so what are the amino acids? As you all know that proteins are the macromolecules which are made up of individual units called amino acids. Right? These amino acids are the building blocks of proteins. That means proteins are the macromolecules and these macromolecules consist of individual units called as amino acids and these are organic compounds. Organic compounds means they are carbon compounds. Right? Now, coming to this particular amino acids, they are characterized by the presence of an amino group, a carboxyl group. And these are the two major functional groups present on the amino acids. So here we have the term amino acids, right? Amino, that is amino group, acid. That refers to the carboxyl group present on the amino acid. So the definition itself indicates that it is having this amino and carboxyl group in its structure. Apart from these two groups, it is also having one hydrogen atom and one side chain. And these four groups are attached to a central carbon atom here. And this is the central carbon atom. And this central carbon atom is referred as alpha carbon atom. Why this is referred as alpha carbon atom? Why? Because whenever a carbon atom is attached to a functional group, it is considered as an alpha carbon atom. And the adjacent carbon atom is your beta carbon atom, the upcoming one is your gamma, and so on. Right? Now here this carbon atom is attached to the major functional group, that is your carboxyl group, so it is referred to as your alpha carbon atom. Clear? So this overall structure of amino acid consists of a central alpha carbon atom which is attached to a carboxyl group on one side and an amino group on the other side. Apart from these two groups, it also holds one side chain and the hydrogen atom. So this is the overall structure of amino acid. Hope this is clear so far, right? Yes. Next, let us see how these amino acids are linked up with one another to form a protein. We have studied that proteins are made up of amino acids, right? So they can't be blindly held. So they should be held or withheld by a certain kind of force or something. So what is the holding force between the two amino acids? These two amino acids are linked through a specific bond known as your peptide bond. Now, what is this peptide bond? So this is a protein chain, and if at all it is formed of a number of amino acids like this, these two amino acids should be held by certain kind of force, and this thing which is holding or linking these two amino acids is your peptide bond. Okay? Now let us study how this peptide bond is formed. How this is formed. So this is your amino acid 1 and this is your amino acid 2. As you all know that this is having one amino group and one carboxyl group. Similarly, the other amino acid is also having one amino group and one carboxyl group, right? Now, the carboxyl group of one amino acid and the amino group of the adjacent amino acid will be condensing or are reacting with each other by releasing one water molecule and initiation of the bond between the carbon and nitrogen. Yes, that means here the carboxyl group of one amino acid and the amino group of another amino acid react to form a peptide bond between these two atoms, that is carboxyl carbon atom and amino nitrogen atom, releasing a water molecule. This kind of bond formed between these two functional groups is your peptide bond. Okay? So the peptide bonds are formed between the amino acids by the release of water molecule. Clear? Yes. Now, hope you all understood what is a peptide bond. And if one bond is there, it is a peptide bond. And if two 2 peptide bonds are there, it is a dipeptide, and if at all 3 peptide bonds are there, it is tripeptide. That means one amino acid is there, this is another amino acid, in between them a peptide is formed. So this is your peptide. And if one amino acid, 3 amino acids are linked up with each other, 2 peptide bonds are formed, right? And this is your dipeptide. Here monopeptide. For mono, just will be representing as peptide. And if at all there are 4 amino acids, the links between them will be 3. Right? This is your tripeptide. So here we can learn that the number of peptide bonds will be 1 less than what? Amino acid number. Clear? That means if at all 3 amino acids are there, then only 2 bonds will be formed. If 4 amino acids are there, it forms 3 peptide bonds. So if a protein is there and we'll say that it is a tripeptide, then what can we say that? How many amino acids are there in this particular protein? 4 amino acids are there. Clear? And how many bonds, or that is your peptide bonds are there? 3 peptide bonds. So the name indicates the number of bonds but not the number of amino acids. Clear? And this is one of the crucial points you should remember in the topic of peptide bond. And that is, for instance, I have told you, further you can learn more clearly. Right? That's up to you. Hope you all understood this one. Next, let us move on to the crucial point and the significant property of amino acids, which is called as zwitter ion formation. Yes. So I think you all know what are ions, right? Ions are the charged species. And now this amino acid also has a capability of forming this charged species, and these charged species are considered as zwitter ions but not ions. Why these are considered as zwitter ions and not simply as ions? Ions are the charged species which will have either a positive charge or a negative charge. Right? If at all it possesses a positive charge, we consider it as a cation, and if at all it possesses a negative charge, we will be considering it as an anion. Right? And if both the charges are present on the same species, right, both positive and negative charge, this kind of ion is your zwitter ion. While we are studying the definition of amino acid, we have studied that it is having 2 functional groups, carboxylic and amino group. In aqueous medium, this will be having a capability of losing 1 proton, and this will be having a capability of gaining 1 proton. Whenever it gains a proton, it attains a positive charge, and whenever it loses a proton, it attains a negative charge. That means here the same amino acid molecule is acquiring both negative and positive charges simultaneously. This kind of ionic structure is called as your zwitter ion, and amino acids are capable of attaining this zwitter ionic structure. Clear? So in aqueous solutions, that is under neutral pH approximately 7, these amino acids exist as zwitter ions we are seeing. But in certain physiological conditions, these amino acids will be existing as zwitter ions at certain pH range. And that pH range is called as isoelectric pH. And this is quite specific for each and every amino acid. And this is particularly called as isoelectric point and is represented as Pi. And we will be studying in detail in the later sessions. But this is the Pi, or isoelectric point, is the pH value at which a particular amino acid exists as zwitter ion. Right? I think this is clear so far. So till now we have studied what is an amino acid, what is a peptide bond, and what is a zwitter ion. Is this clear? And the main point regarding these amino acids is the amino group, carboxyl group and the hydrogen atom will be seen for all the amino acids. And the only point where these amino acids will be differing is this your R point. Yes, and this is the side chain which differentiates from one amino acid to the other. Depending upon the side chain, we can categorize these amino acids as specific amino acids. Clear so far? Yes, hope so you all understood this concept, and thanks for watching. Thank you. See you in the next video.